Arts. I'm the president of RCA, and welcome. And we're so glad to see so many people here this afternoon. Please make sure you do get refreshments as you want them. Please eat them up. They are delicious. Let's see. It's my pleasure to introduce Dr. Stephen Rich. We actually had him lined up to come a couple of years ago, and then, as you all know, the world kind of shut down and we had to postpone for a while. We were finally able to get him back, and we're thrilled. As you all know, ticks are a problem in this area, and Lyme disease. I think most of us know someone that's been affected, and so it's great that we have some current information to share today. So thank you very much and welcome.
Well, thank you. Thanks for the invitation. So this is a beautiful spot. I was remarking how this is not what I think of when I think of a field station. I think of dirt floors and, you know, exposed timber. This is a beautiful place. And I grew up in upstate. I grew up in Watertown, and so I've been visiting family. So I didn't drive all the way from Massachusetts today. I was with at my mom's house last night. So it's good to be back, sort of coming back home. I'm going to do something a little bit different in terms of presentation. So rather than a PowerPoint, I'm using this program called Menti, which for professors or instructors, you might find this kind of a cool thing. So I'd encourage, if you would, just go to, if you have a smart device, phone, whatever, if you go to menti.com and use that code, you can participate. You can sort of, there's interactive slides like this one. And it's nice because I do it with I have a 300-seat gen ed course that I teach. And it's the only way I know to like really have an engaged conversation when there's 300 people in the room. And there's a couple of other reasons that I like to use it. So if you go to Menti, you don't have to download any software. It doesn't take any information from you. You just enter that code, and then you can answer this respond to this question by telling me what ticks make you think of. Let me go to the next slide, and it's a little bit easier because you don't have to type anything in, but you can express how you're feeling today. You know, batteries are charged, you strongly disagree or disagree with that statement, excited for spring, worried. So if you want to do that, and the reason I think this is good is what I've sort of experienced doing. So for now, I guess 6 or 8 years now, I've been going out and giving public talks. I'm gathering most people from the university here, most people from Sweden. So it's a little different audience than I was anticipating. I was thinking it might be a general audience. What I experience is that you go out and people hear you're coming to talk about ticks, and I've got things I want to explain about ticks. But most people come in with a series of questions that don't necessarily line up with what I'm going to say. So I think it's better to have this be a little bit interactive. I do have a few things I want to tell you, and then by all means, just interrupt me at any point and I'll answer your questions. There's also like a way to sort of interact with this thing and answer questions. But for me, this is a way of getting out of what I call the death by PowerPoint mode, where you just like stand and you just give slide after slide after slide. Because again, a lot of you might already be scientists, but the general public always seems to be frustrated by the fact that they're trying to write things down furiously that I'm saying in a lecture because they want to know what to do to protect themselves. You don't need to write anything down furiously. If you just listen, hopefully I can tell a story and I can put some comprehension in your hands rather than just communicating information. So rather than death by PowerPoint, I'm going to do this interactive thing. Okay, so here's sort of my title slide. I'm going to talk about tick-borne disease risk and response. Risk, my funny play on words there, and that's who I am. So these are some of the things that I've learned on these little junkets going out in the public. It's what the general public view is. Let me minimize that so you can see. That lived experience matters. So the first thing I notice when I go out and I tell people ticks don't live in the grass. You won't get tick bites in the grass because ticks don't exist in the grass. And everybody will come back and say, I got bit by a tick in the grass. Well, the predominant place that you find ticks is on edge. And I don't want to ever spend I learned I don't want to spend a lot of time trying to argue with someone's lived experience that they sat down in the grass and they got a tick bite. I'm telling you that most of the ticks are on the edge, less in the grass. So lived experience matters. And a lot of what I'm going to tell you might not conform to your lived experience, but what I'm telling you is what the best science does. The other thing I know is that people are looking for simpler solutions. So again, that's sort of a general public thing. People say, I want to know what to do and I want to know when to do it to be tick safe. And unfortunately, this doesn't lend itself to those kinds of solutions, this particular problem. And we have other problems that we're experiencing in public health right now that are similarly complex, right? It's not like one vaccine takes care of all the problems, or one public health measure takes care of all the problems. These are management issues. And so hopefully I can help you to understand or share with you what I understand to be the science of it, so that you can understand why we do what we do as much as what it is that we do, and then you can make better decisions. The last thing I just want to say is that a pervasive thing that I've seen in the past, I guess almost a decade now that I've been going out and doing these public talks, is that fear is the pervasive response that people have with the ticks. And none of us makes our best decisions when we're afraid, right? So I want to try to push out the fear and replace it with knowledge so that we make informed decisions. And I also mention that because I would be wary of anyone that tries to instill fear in you about ticks and tick-borne diseases, because there are lots of things that you can do to vastly improve your likelihood of protecting yourself or securing that you're not going to suffer from a debilitating disease. Unfortunately, I think fear mongering is prevalent in this field, but I would always be wary of that. I'd be wary that and be advised that there are things that you can do to drastically reduce your risk. So let me start by asking this question. So this one is interactive. I have two people up there. Tell me which of those two people on the right or left, him or her, pose to you, if you were in the room, this is an airport or something, poses the greater risk to you of COVID. So, like most people that would be given this question, you're cueing on him as posing the greater risk than her for a couple of reasons. One is he doesn't wear a mask and she does, right? I mean, that's the obvious one. The other is people might think that there's something to do with the phone, like he's on the phone. Maybe that says something about something, who knows? What I would argue is that you just don't, I have not given you enough information to make that assessment, because you don't know whether they've both been vaccinated, whether either one of them are infected. We don't know, right? So what we're doing there is we're mixing up the threat with the risk, with the exposure. We're not clearly understanding where the real risks are. So what I want to do is to use that sort of an example to build out and for us to think about how we can think about risk of tick-borne diseases. And then I want to tell you some things about tick biology that can help you to reduce that risk. So one more example outside the tick world, just to keep sort of press your intuition into this. If you imagine you're getting ready to go in the water and you're thinking about the concern about sharks, here's another example where, look, there's two different scenarios where there's sharks in the water, but the risk to these people differs between the left and the right picture. This is a low risk situation where the person hasn't entered the water, and this is a high risk situation. So speaking to that point a moment ago, we didn't know whether the threat was present because we didn't know if either of those people had COVID, but we were making some assessment of risk. In this case, we know that the threat is present in both cases, and what's differential here is the exposure, whether we actually expose ourselves to that threat, right? So let me turn that question now. So is that tick, which hopefully the picture's visible, you can see clearly, is that a risky tick? Does that tick pose risk? Same question that I asked about the couple before. So to think about that for a minute, let me ask the question a different way. Are all ticks risky? What do you think, yes or no? Yeah, so I would argue that not all ticks pose the same risk, and some ticks pose very little risk. This tick probably poses a slightly higher risk than another tick that I'll show you in a moment, because it's a female deer tick and it has a higher likelihood of being infected with a pathogen than other species. So I would argue that this is probably a little bit riskier tick than if I showed you another picture of a tick. So you can learn something just from the tick species about its potential risk. And what we should be thinking about is that risk is what you read about in the newspaper or you see from the CDC. Risk is the incidence of disease in people. So this is done from clinical reports of who's getting sick over the total number of people. And then the other thing that you'll see often is assessments of hazard. So you'll hear scientists that go out and collect ticks in the wild, and then they test them for pathogens and they tell you the infection rate. But what you're really interested, what you really should be queuing on is the exposure, which is the two pieces that bring that together. So it's not just about how many people are sick, because sick people don't beget sickness in other people. It has to go through the ticks. And it's not just about the ticks in the wild, because a tick in the wild that never bites a person poses no risk at all. So what we really need to be focusing on is the exposure. How ticks bring the pathogens that they have into contact with humans. So I'm going to tell you the three things that are fundamentally important to understanding that exposure risk. The three things that you can know that can drastically change your exposure to your experience with ticks. The first one is to know the tick species. So as I alluded before. So there are three principal human biting ticks in North America, three species of ticks that bite on people. There's dozens of other species, including the Asian longhorn tick, which is a newly arrived one, but they generally don't feed on humans. There are woodchuck ticks, there are squirrel ticks, there are groundhog ticks, a bunch of different tick species, but really only three that feed on people. So another question, which of those ticks is associated with Lyme disease? And you can just click on your phone, you're playing along, you can click on your phone and say which ones are associated. Excellent. So you all know that these are deer ticks or black-legged ticks. These are dog ticks and these are lone star ticks. And they are males, females, larvae, and nymphs of each species. These species are not associated with Lyme disease whatsoever. They do bite humans, but they're not associated with the pathogen that's associated with Lyme disease, whereas these species do. The predominant individuals that transmit it are believed to be the nymphal form, not because they have a higher infection rate, but because they're smaller and they feed for a longer period of time. And in that regard, they pose a higher risk, which I'll speak to in a moment. But males and females can both be infected. Males tend not to eat or not eat very long. They take very small blood meals while they're seeking females to copulate, and the females will take a fuller blood meal and then lay their eggs, about 2,000 eggs per generation. Larvae are actually, of this species, are not infected because there's no what we call transovarial transmission of the pathogens. So Lyme disease pathogen has to be acquired in the life of an individual tick. A tick gets infected when it feeds on mostly an infected white-footed mouse. So larval ticks, by definition, if this tick gets on you, it is never fed before because they only take one blood meal in each of those life stages. And if it's never fed before, then its chance of being infected with Borrelia or Lyme disease spirochete is vanishingly small. Okay, so the riskiest ticks are these guys. These are also posing a risk, although this one doesn't feed very much. And these species are not associated with the pathogen whatsoever. So that's just a shakedown of it. So there's the labels of the species. There's deer ticks. Deer ticks or black-legged ticks, dog ticks, and lone star ticks. And these are the list of pathogens. So first thing to know, or next thing to know, is that it's not just Lyme disease involved here. So Lyme disease is one of the pathogens associated with these ticks. It's associated exclusively with this species, but there are other pathogens, including Babesia and Anaplasma, Babesia being a malaria-like illness that's transmitted by those ticks. Anaplasma is another bacterial disease. Borrelia miyamotoi, which is a very different Borrelia species that lives in the resides in the blood, and then even some viruses like Powassan or deer tick virus. And while these ticks are not associated with Lyme disease, they are associated with other pathogens, although those are somewhat rare, and I'll show you figures for that in a second. So things like Rocky Mountain spotted fever associated with dog ticks, not black-legged ticks. Starry, which is a little bit of a mysterious disease. We don't exactly understand what the pathogen is, but that's often associated with Lone Star ticks. And then this really odd thing called alpha-gal syndrome, the red meat allergy that maybe you've heard about. That's associated, at least for the moment, with Lone Star ticks. And I can talk about that more in a moment. But for a moment, I just want to go on with those three things that that are the predominant risk factors. So let me show you what a tick does in its livelihood and what it doesn't do, I guess, as much as anything. So here's a young lady walking through the woods, and here's what ticks do. And I tell you that they don't fall, they don't fly from trees, excuse me, they don't fly, they don't fall from trees, and they don't jump. What they do is they crawl out to the end of a branch, and then they quest. So they put their front legs up, and they wait for some unsuspecting individual like that young lady to come by, and then they grab on. And then they crawl on that woman's body until they find a place. And sometimes they can stay on for several hours. Sometimes they might stay in your clothing and bite later in the day. But eventually the tick, is going to find a place to drill down. It's before dinner, right? So it's okay. Yeah, so then she drills down. She actually cements herself in place. And then she starts this process of spitting and slurping, spitting and slurping. And that's what makes ticks such exquisite disease vectors, is that they have this very intimate association with the victim they're biting, and that they're both taking blood out, but they're also spitting saliva into the side of that wound. So what's in the tick ends up in the person, and what's in the thing that it's feeding, or the host or the person, ends up in the tick. So that intimate relationship makes them ideal vectors so that they can move the pathogens around. So what can you do to protect yourself against that? Here's where those things come in, like what can I do? When do I do it? Bearing in mind that ticks aren't like mosquitoes. Like a mosquito on this side of the room can smell you and fly to the other side of the room and try to bite you. Ticks don't operate that way. They use that questing behavior. They go out to where they think a host is going to come by, and then they wait for the host to come by, and then they grab on. So the typical ways that we think of, for example, using spatial repellents or contact repellents, don't work the same way against ticks. The way DEET works to protect you against a mosquito bite is it basically masks the mosquito's ability to recognize you. So basically it's clouding the mosquito's vision so they can't find you, they can't land on you. Well, there's nothing to be clouded. Think about that situation with the woman walking by. There's nothing, the tick's not cueing anything, it's just waiting for her to come by. So there's no opportunity to be repelled. It's just going to grab onto what comes by. The way repellents work is against ticks is their contact repellents, and the ticks are literally repelled against the repellent that's on the skin. So for example, I'm getting a little bit ahead of myself. Before we get into repellents, let me just say that there's a couple of things you can do independent of any chemical treatments. You don't necessarily need to use repellents or acaricides. One of them is you wear the proper clothing. So there's this fashion statement of tucking your socks into the inside, or your pant legs into your socks. And the reason for that is those ticks, although that one I showed you before was an adult and was fairly high in the vegetation, very often in the nymphal stage, they're very low. So a tick takes its purchase on a person probably around ankle height or lower, and then it crawls up seeking a place to eat. So if the person has their pants bloused such thusly, then the tick will crawl up on the outside, and there's a chance that before it gets to the good parts where it wants to bite, it will either fall off or be detected and be pulled off. So the whole notion of this is just basically to thwart the tick's ability to get to your exposed skin. It's not 100%, but it does seem to work pretty effectively. The other thing is light colored clothing, not because ticks have a preference for color, just because the dark ticks are easier to see against the light clothing. And then a big thing, which is going to be an emphasis, is to always check for ticks, because you'll see in a moment, a lot can be done in terms of disease prevention just by taking the tick off sooner rather than later. I don't know why the arrow's not working today. Okay, so what can you do for personal protection? This is the part of that talk where I run into people's lived experience. What I can tell you is that there's two approaches that are sort of tried and true. One is permethrin treated clothing. So permethrin is a acaricide, not a repellent. So it doesn't repel ticks or push ticks away. It kills ticks on contact. They contact the chemical through their feet. And you treat your clothing, not your skin, just because it's engineered so that the material adheres to the fabric and then gets into the tick that way. So the ticks crawl on the permethrin treated clothing. They absorb the chemical through their feet and it kills them. So you think, okay, that sounds like a poison, and it is, but its safety profile is very, very good. There's the issue with the, oh, sorry. I thought maybe I had a problem with the Zoom. Everything working okay back there? Great. Oh, should I stand in front of that picture? Is mine moving around? No. Okay, all right. Permethrin. So, yeah, so it's a very effective, so it's has a very high safety profile for humans. It's something like 2,000 times more toxic to ticks than it is to humans. Has a very good safety profile for us. Good safety even for children, although not the smallest of children. It is toxic to cats. So in a liquid form, it's not recommended, but for the most part, it's a pretty safe as far as chemistries go. And then DEET, of course, is a repellent, so a different category. This you don't apply to clothing, you apply to skin. So you can demonstrate this, you can demonstrate on clothing or on skin, that the ticks will crawl to the area where it's treated, and then as they approach the treated area, it's almost like their feet are burning and they'll move away. So we can do this in wall climbing tests. Ticks have a tendency to want to climb up a vertical surface. If you set up a vertical surface with a line of DEET, they'll crawl to the line and then either crawl away from it, or they'll fall off when they reach the line. So both of these are pretty effective treatments. There are other, in some cases, non-synthetic, so-called natural compounds. Most of them, with the exception of maybe picaridin, most of them, at least based on the best science, don't seem to be as effective as either of these two mechanisms. So cedar tree oils, essential oils. And again, people will say, I wore grapefruit extract or whatever it was, and I never got bit by a tick. Can't say that you would have been bitten by a tick if you weren't. Can't say that it's working or not. Just telling you what the best science is, that those things don't seem to be, when done, exposed to the same rigorous scientific processes, don't seem to be as effective as these mechanisms. Anybody want to shout out about that? Yeah.
Quick thing about
Sure. Yeah.
It seems on the bottle that it's dangerous when it's in a liquid form, and then when it dries, it's not much of a problem for anything.
That's right.
Except ticks.
And it's really a problem for cats, as I said, in
Yeah.
a liquid form.
In the liquid.
That's right.
So hang it up, get it dry, and then you're good to go.
Right. And it's probably as much as anything.
Don't wear it wet.
Right. And it's not the permethrin probably as much as the solvent that's volatilizing off as it dries. So that's the reason for the toxin. Yeah.
So putting DEET on clothing isn't effective?
It's not effective because it doesn't really last very long. It doesn't and that's a downside of DEET against ticks on your skin, is you have to keep reapplying it. So that's the main reason we discourage people from putting on the clothing. Yep.
You said not young children. Is there a reason for that?
Usually,
They're more susceptible to any chemicals.
just more susceptible to chemicals in general. So yeah, under 2. Yeah. Anything else? People want to argue for cedar tree oil or any of these? So it's quite common. I mean, we hear it a lot so ticks are relatively new here, right? So if you all have lived here for a while, you know that there weren't ticks to speak of 20 years ago. 20 years ago, Cape Cod, they were knee deep in ticks even then. And so in the lore, these things are much more prevalent, the cedar tree oils, and I use cedar tree oil, but lots of different essential oils. Oops, sorry, went one slide ahead. So the other thing that you can think about as a control measure is source reduction. So there are things that you can do to protect yourself, either personal protections with acaricides or repellents or the clothing, or you can try to reduce it at the source. So again, this is, well, I know I noticed there was some talk about doing this at Fort Drum some years ago. You can either kill the questing ticks, so perimeter yard sprays, which doesn't seem like they have very long-term effects. So the idea is You've got, your nicely manicured lawn, then you've got the edge leading into the forest that's behind your house. If you do a perimeter spray around there, you can create a temporary border that prevents ticks from coming in. What it doesn't prevent is a white-footed mouse from coming a couple hundred yards across that line and bringing it to the other side of your property, because they're not similarly, it's not a barrier for them. The other thing it doesn't do that sort of creates a false sense of security is, while your yard might be safe, if you cut through your yard to go to your neighbors for a barbecue, you just exposed yourself, because you basically are still going into harm's way, as it were, because you're outside the barrier. So again, a lot of people spend a lot of money to do barrier sprays. There's no evidence that it's terribly effective in terms of long term. The other thing you can do is you can try to kill ticks on hosts. So the idea here is to reduce the total number of ticks. So if you could caricide treat white-tailed deer, which is the principal adult host of the ticks, that you can reduce the tick numbers that way. Again, this is probably like a numbers game. One tick can lay 2,000 eggs. One deer can feed, I've pulled upwards of 3,000 ticks off a single white-tailed deer in Ipswich, Massachusetts. So just trying to control ticks by killing ticks on deer, it's a kind of a losing battle. Like, we're not going to be able to eliminate them all. And places like, right now, there's on Martha's Vineyard in Nantucket, there's usually about any given time about half the population that wants to extirpate the deer from the island. The other half wants to protect Bambi. But what we always tell them is, even if you were to reduce the deer herd, say 80%, you would have the same deer density that they have on Cape Cod, which is the mainland, and they have a huge Lyme disease problem there. So it's not a way to eliminate the problem. It's not a sterilizing technique. Do you have something to say, sir? Okay, sir. And then you can reduce or modify the host. So you can also do things like, again, on Nantucket, there's a MIT scientist that wants to release transgenically engineered white-footed mice that are refractory to infection. So white-footed mice are the principal wild reservoirs of Borrelia burgdorferi. He wants to make a mouse transgenically that can be released in the wild and won't be able to be infected with Lyme disease, and will break the cycle that way, which I think is a bit of a Buck Rogers kind of approach to the problem. And there's a couple of things wrong with it. One is, well, first of all, we haven't even proven that this can work in the lab yet. Secondly, it would only protect against Lyme disease and not the other pathogens that I showed you on that list, or against other tick species. So it's one of the tools out there. It's probably not it's not going to be a final solution. Okay, so I told you there's three things. First thing was know the tick species. The second thing important to know is how long the tick is fed. That's the second very important thing, to understand the risk associated with a tick that bit you. Why is that? That's what ticks actually look like. I showed you the nice pictures that you'd see, like, well, we have these little ID cards that we distribute as well. But if you look at a pamphlet or a brochure and you're trying to figure out what ticks look like, you see very nice pictures. This is what ticks look like after they've been on a dog or a person or you for any length of time. They get mashed up. This is a tick with its mouth parts missing and its scutum ripped off. Here's ticks that are partially fed. They change their body shape. They change their pigment, their coloration. They all look very, very much different. But what I wanted to also show with this slide is that the risk of transmission of disease increases as these ticks feed. So a tick that hasn't fed very long poses less risk than a tick that has fed for a long time. And I'll show you why in a moment. And the reason I go through the trouble of trying to show this to people is that very often, again, it's one of those lived experience things where people say, I had a tick on me for 5 minutes and I got Lyme disease. Can't understand how that happened. All I can tell you is how the biology works as we understand it. I would say that very likely in those scenarios that someone got bit by a tick for 5 minutes and got Lyme disease, they got bit by another tick on the other side that they never saw, that fed for the length of time that we think is necessary on average for the pathogens to transmit. Also, the estimates for transmission times are they're estimates, right? So they have a normal distribution. So while we say that the average time is probably upwards of 48 hours for transmission of Borrelia burgdorferi, that's not to say that you can't get transmission occurring in 12 hours. I'm just saying that it's much more likely to happen at 48 hours. But let me speak to the biology of it. So here's why we understand that Borrelia burgdorferi only gets transmitted after 48 hours. When a tick feeds, so this is that similar picture as we showed before. So here's the surface of the host. Here she has cemented herself into place, and she's got her mouth parts into the feeding lesion. When she feeds, and all I've done is, this is bilateral, but I've just shown one side. I've shown the intake and the other side, the outtake. One side, she's got her midgut. This is where the blood comes in. And the other side are her salivary glands. That's the spitting. Remember I said before that they slurp and they spit, they slurp and they spit, they slurp and they spit. And this is a recurrent process through the ticks can feed for 6, 7, 8 days on a single host.
And is the spitting part of the anticoagulant, or why is that?
Yeah, it's part of an anticoagulant and it's part of the digestion. They're cultivating their blood meal in that feeding lesion. So the digestion process is taking place outside the tick's body to a certain extent. But what I wanted to emphasize here is that these are only one-way streets, that what comes into the tick from the host into the tick goes into the midgut and cannot go back into the salivary glands. So for example, ticks, one of the myths, ticks don't regurgitate. Ticks can't be forced to regurgitate or squeezed anything out. They actually have pharyngeal blocks that prevent that from happening. The only thing that's coming out of the tick is basically coming from the salivary glands. So pathogens, if I were to animate this, if this pathogen came out, this pathogen is going to end up in the midgut of this tick. And to move from the midgut into the salivary glands, it does a very odd trick. It turns out that Borrelia, this very minimal parasitic, the small parasitic genome, they don't have the proteins, they don't have proteases to cut themselves out of the midgut and into the salivary glands. They use the serine proteases from the blood itself of the next host to co-opt and basically chainsaw their way out of the midgut and then back into the salivary glands. And all of that takes time. All of that requires that they take that second blood meal 24 or probably closer to 48 hours before they get accumulate enough of that serine protease that they can cut out of the midgut and get back into the salivary glands and come out. So two things. One is intake this way, outtake that way, and they must have blood in order to get back into the salivary glands so they can get to the outtake, the outgoing traffic, as it were. That's why pathogens take several hours or several days to transmit. Now, it differs for different pathogens. For Borrelia, it's about 48 hours. That's the Lyme disease pathogen. For one of the other pathogens, Anaplasma that I mentioned, it's probably closer to 24 hours. And then for Powassan virus, which your colleague here at Syracuse University studies, for Powassan virus, the thought is it might only take 15 minutes. So some things can come out much quicker, and probably in the case of Powassan virus, the reason it comes back is that it's more of a mechanical transmission that the virus retains in the mouth parts. So when it's feeding, it doesn't have to go through the salivary glands, into the midgut and the salivary glands. It can transmit quicker. The exact mechanisms aren't understood. The exact timing, I think, still remains an open question, but the estimates now are that that takes about 15 minutes. The point being that none of these pathogens are instantaneous, that none of them see transmission from a tick into a host in an instant. So for things like Lyme disease, as we understand quite well, as a tick gets bigger, the risk of acquiring Lyme disease from that tick increases. So these are nymphal ticks along the top, days of feeding. These are adult ticks along the bottom, adult females, days of feeding. By the time you get past basically 1.5 to 2 days or 2 days, everything this side poses risk of whatever's in that tick getting into you. So the good news is, if you pull your tick off, we were talking earlier about people pulling ticks off themselves. If you're pulling your tick off in these days, you're greatly reducing the risk associated with that tick. You're doing a lot to protect yourself. I don't think I have a slide to say this, so I do want to say something about taking the ticks off. There's no magic way to take a tick off. There's a lot of there's lore about, don't take the tick off because you might squeeze it and squeeze the contents back in. Doesn't work that way. Because, as I pointed out before, because it's a biological process and the use of the serine proteases, the tick has to be alive for the Borrelia to get out of that tick and back into you. So if you pull a tick off and you rip it in half and kill it, and you leave part of it behind, that tick is not transmitting any longer. What that tick might pose to you is the risk of, just like you have anything else in your skin, you could get secondary infection. So you want to clean that. And maybe you want to spend a little bit of time like trying to pull out those pieces, but it's definitely not recommended that you go digging around and scrap and trying to cut those pieces out. Surface antibiotic, sterilize with some alcohol, wash it with soap and water and treat it like you would a wound, and it'll clear. You've done a lot to protect yourself that way. Don't use a burnt match. Don't put Vaseline on it. Don't put nail polish on it. There's a whole bunch of different ideas that if you put these substances on or expose it to heat, that the tick will back out. She's not going to back out. She's invested. That's the only blood meal she's going to take in that life stage. She's glued herself and she's ready to stay there. No amount of chemical or anything is going to make her back out. She's there. So you have to pull her out. And anything that you do that leaves the tick live and attached to you longer only gets you further along in this risk profile. And so you're not doing yourself any good. So even if you're out in the backcountry and you get bit by a tick and you don't have your fancy tweezers or tools to pull them out, get your fingernail, get as close to skin to skin and pull it out, because every hour that you leave that tick on increases your risk. Any questions about that? Yes.
The one that's gotten as big as day 7.
Yeah.
Do they just fall off naturally then?
They do. So what she does in the natural situation is this would usually be on a white-tailed deer. And shortly before falling off, she would be mated, and then she'd fall off into the leaf litter and then lay her eggs. 2,000 of them.
I have a question.
Yeah.
So right now, just looking at this as a representation, we could fixate on that. Well, that engorged tick at 7 is pretty scary. But really what we're dealing with is something that's microscopic. And that's where the sophistication and say the transmission is happening. And you're going to probably come to that at the end, but I think it's hard to see that when you have these kind of representations in front of you. You have the mechanism, let's say, that as opposed to the unseeable that's acting. The point, no real point, it's just like a matter of perception.
Yeah. So when you say microscopic, you mean the bacteria that are inside it are the actual risk factor. Yeah.
So it's something I'm always trying to work through my head. It's like, well, we're really dealing with a pathogen.
Yeah.
We're not dealing with a deer, as a singular thing. But
Right.
Ultimately, if you boil it down, it's a pathogen, and that's what we're looking at.
Right.
Just something you said.
Yep.
No
No.
real point,
Yep.
and an observation.
Nope. Thank you. Anything else? Yeah.
I don't know if it's a question that thrown in. The availability of pathogens is different in different environments. So different parts of the country may have the same animals, but haven't developed the pathogen load. It's say someplace like Connecticut or Rhode Island.
Well, what's curious,
Is that right, or is that?
what's curious is wherever we see black-legged ticks proper.
Yeah.
So and when I say proper, the ticks in the southeastern United States don't seem to be the same species of tick.
Okay.
But wherever we see black-legged ticks proper, like places like Watertown, New York, where I grew up, where they weren't 15 years ago, they're loaded with Borrelia. The adult females are 50% infected with Borrelia burgdorferi, the same as Cape Cod, the same as Chittenden County, Vermont, the same as Aroostook County, Maine. Wherever the ticks are, they have that transmission rate. It's probably
Makes them a sufficient vector themselves, right?
right. So they're probably moving. The reason they get moved up there is they're moving on migratory birds, And then they lay down on a bed of white-footed mice, which are competent reservoirs, and the transmission cycle gets set up anew. And it's because they're very effective at feeding on white-footed mice and white-tailed deer as adult hosts. Yeah, so that was, your intuition was exactly what I was thinking 20 years ago. When I was a new assistant professor, I got some money from NIH to go out and look at some newly emerged areas, thinking, oh, maybe we'll see like a founder event. Like there'll be very low transmission rates, and maybe we'll just see a couple of different genotypes. Maybe we'll see what Lyme disease looks like when it's nascent, when it's just starting out. Everywhere we went that we found ticks, it was there. So either, I mean, there's a chance that we're not getting there at that very instant when it's exploding, but we never found such sites. Yeah.
So are white-footed mice, the common mice, I don't know, that's just the mouse, when you see it running down the basement, that's probably.
If you've got a mouse like that's in your attic, it's probably white-footed mouse. Anything else? All right. Thanks for the questions, by the way. That's very helpful. All right, third thing. I told you there were three things. The third thing is the infection status of the tick, because not all ticks are infected. So to your point, it's not a tick, it's what's in the tick that's dangerous. So with our example before, we didn't know whether these people were infected, although we were guessing that he's more risky than her. We don't know if either of them that's infected. Similar analogy here, like a tick gets on us, even if it's fed for 48 hours and it's not infected, it's a drag, but it's not necessarily exposing us to anything. So here's the numbers. So black-legged ticks, American dog ticks, Lone Star ticks, and then a sampling of those pathogens that I showed you before. This is the biggie. Borrelia burgdorferi is about 50% in the adult ticks and roughly 30% in the nymphal ticks. And there's a reason for that, because they only acquire infection when they feed on an infected host. These ticks will have fed twice and have twice the chance of getting infected, whereas these ticks have only fed once, which is why there's roughly a twofold increase. Things like Babesia, that malaria-like pathogen, 5 to 10%, again, differs a little bit. Western Massachusetts is a little, I know Mass just a little bit better because we have field sites there, but Western Mass a little bit lower than Eastern Massachusetts. My guess is that upstate New York is lower still. I haven't actually looked at any data there. And then Nanoplasma Effect is a tough one, also 5 to 10%, although we can go to certain parts of the country like the coastal islands of Massachusetts, and you can see upwards of 40 or 50% in that one for reasons that we don't entirely understand. These other ticks, albeit associated with different pathogens, you notice that the rates of infection are much lower. So here again, these are the principal risky ticks. These are relatively low. It's not a 0%. And Francisella tularensis, if you don't know, this is tularemia. That's a nasty infection. Can be both the surface of the skin, or it can get respiratory as well. Can be very high fatality rates. Ehrlichia chafiensis, also not a cakewalk, but relatively small infection rates relative to Lyme disease ticks. So you can learn a lot, as I said before, just about your risk by knowing how to differentiate the tick. But you can take it a step further if you actually can look at the infection status of the tick that bit you.
Is that also associated with rabbits?
Yeah. So it was called rabbit fever, I think, because rabbit hunters would often get it. They'd be butchering the rabbit and they'd slice, cut their own skin and get an infection that way. It's not unlike anthrax in that it can infect the skin and cause these bulbous surface infections, or it can be inhaled and cause dangerous problems that way. So what I'm going to tell you about now is just tick report, so that I have a disclaimer to give you here. So tick report is our way. So this is something my lab started a few years ago, 2006, 16 years ago. Where you can send your tick and we can do the assessment of those 3 different characters. It's a fee for service. You place the order, you mail the tick in, and then you get results in 72. Actually, we got that down to 48 hours. This is my disclaimer. So I did this for many years as a professor at the University of Massachusetts up until 2020. And it got to the point where the university, because of COVID, was doing shutdowns. We couldn't do the service anymore. And so the university encouraged us to actually move it off campus. So we created a company called MedZoo Inc., which is a B Corp. I don't know if anyone knows what B Corps are. I didn't until we created this one. A B Corp is not a nonprofit. It's a entity that has a benefits mission, which supersedes its profit mode. So basically, we've written to the DNA of the company that we're going to do public health good over profits. It does generate profits that basically goes to pay the salaries of the people that do the testing, but it's no longer a part of the University of Massachusetts. I'm here today as a professor at the University of Massachusetts, and so I'm not necessarily, well, I just want to disclose that I have this financial interest as a principal owner in MedZoo Inc. that now does the tick reporting. So that's the process. You send the tick bite in, you order the tick report. We do the 3-point assessment, the 3 things I pointed out before, and then the results pointed to you. The other labs that do this, Saravan and Thangamani at SU does this. He does it a free service for New York State residents. Connecticut Ag Station does it as a free resident for Connecticut residents. University of Maine has a paid service. We were the first ones to start doing this, and it was, I guess, such a good idea that lots of other people are doing it as well. That's what a tick report looks like. This is actually a tick that bit me. I broke it off when I was removing it, so the back parts were missing. It was positive for Borrelia burgdorferi. I elected not to treat. You could treat prophylactically at that point. I knew the tick hadn't been on me very long, so its infection status were not a principal concern for me. I continued to watch the site of the bite for any telltale signs of the rash. I never got it, so I never got treated. And I've never tested positive. I've never had a positive Borrelia titer. So with tick report or tick testing services, we argue, and I'm not going to say much about this until maybe the very end, I'll throw out a little bit on this, but we argue that there's benefit to individuals to know whether the tick that bit you is positive or more risky based on those 3 different factors. But what we think is equally or maybe more important is the back end of this, that when you put all of that information together, it makes for a very nice public passive surveillance network. So when we look at the aggregated data of we test about 16,000 ticks, 16,000 human biting ticks per year, we can get information that allows us to assess the risk to populations by looking at the risk to individuals. So let me just share with you a little bit of that data. We can answer questions like who's getting bit. Here's the answer. So this is actually from a paper that we also make all these data freely available on tickreport.com/stats. And then we periodically publish in peer-reviewed journals. So this was a peer-reviewed paper, I think, from 2014. These are Massachusetts data. This purple line is the incidence of Lyme disease across age groups, less than 5, 5 to 9, 10 to 14. And then these little bars indicate the total number of ticks, number of nymphal ticks, and number of adult ticks. And you see there's this bimodal distribution, both in the ticks, but also in the incidence of Lyme disease. So you may say, big deal, doesn't surprise me that they would match up. But until we did this, we really didn't know why there was this dip in the group of people 19 to 30, other than the fact that we knew they had less Lyme disease, but we didn't know why. We now know that they're actually getting bitten by ticks less frequently. We don't know why that is, whether it's that they spend less time outdoors or somehow are more resistant to tick bites, we don't know. But this was sort of a nice way of getting public health information. So this is that risk information I talked about before that comes from the Department of Health, or this is the risk information. And we can see how the passive surveillance data reflects the same kind of pattern. We can also tell when people and their pets are getting bitten. So this is just 2017 data. So the bars are for deer ticks, dog ticks, and Lone Star ticks. These are data for the state of Massachusetts. We actually can break this down by zip code, by county, although obviously the density is not the same, but we can do things like we can show people firsthand. Well, here's one of the patterns that I always, when I'm doing talks in Massachusetts, I point out that there are 2 peaks for deer ticks. One is in the spring, about this time of year, and then another one in the fall. And the reason I point that out is whether I'm like at one of my kids' hockey games or whatever it is in September, I'll see people and they'll be like, oh, ticks are there's really low numbers of ticks this year. They're talking about September. What they're doing is mistakenly comparing September of that year with June of that year or June of the previous year. They're not noting that this dip occurs every September. So every September I'll run into people and they'll say, well, the ticks are really good this year. I said, nope, the ticks are really good right now because they're down right now in these months of August and September. The other thing you can see is that while deer ticks have the 2 peaks, dog ticks and Lone Star ticks have just the single peak. They tend not to feed on people as often as the deer ticks. And if I didn't say, all of these are human biting ticks, so every one of those ticks came off an individual person. There's 2018, you see similar patterns. We do see a little bit of variation. So like the New York State, especially upstate New York, it tends to start a little bit later, although not statistically meaningful as yet, but it's probably the winters are a little bit ending a little bit later up here. Yeah, yes.
I just had a question about a couple slides back, the images of the up and down. When you were talking, you mentioned how like
That one?
down, no, back in the 2. That one, yeah. You mentioned how it goes down as people get closer to like 20, 25, and then it goes back up as they approach 50. Would that be because that's the age people start having children? Because they're high in like young children, and then they increase in adults around a certain age. So would it be plausible that adults with children who are out in the woods are also more likely to get ticks because they're also like in the same areas as these kids?
It's plausible. I don't know that that's an explanation, but it's plausible.
Do you have an option, again, to the same graph, for like I went outside today and I didn't get hit by a tick because I feel like you have to compare those statistics in order to determine the fact that you're not just facing a lack of representation rather than a lack of actual tick bites.
No, you're right. There are sampling issues here, not the least of which is maybe these are people that pay for the service. So they pay the University of Massachusetts to have it tested. Every person on this graph did so. So it could be that these people just have less disposable income to spend on a tick test. So that could be a sampling effort. And yeah, we don't know if there's something about this group of people, maybe they just don't, even if they have the money, they don't bother to send a tick. There's lots of, there are some sampling issues. Does that answer your question?
Yeah.
But very astute too. You're on the right track with that. Just to finish these out. So this is just showing the underlying. So those last ones were the 2 different species. This is just for the deer ticks, and it just shows you that there's 2 peaks of adults, 1 peak of nymphs, and a very low, the little blue bars here are the larval ticks. We don't know exactly what's going on there. We don't know if it's just that people are missing the ticks because they're very, very tiny. So when you hear people say it's the tiniest ticks, these are the tiniest ones. And whether the people are just missing them, we don't know. We've even offered sort of a bounty where we said, we'll test any of these ticks for free because we really wanted to see whether people are getting bit by larval ticks. And still people they're not sent into us in that period. So whether those ticks have a much lower attack rate on humans or not remains an open question. Remember, those are also the ones that are not infected. There was actually some work done by some of my colleagues at Tufts University where not wild-caught larval ticks, but laboratory reared larval ticks were being used to do xenodiagnosis. So the best way, I didn't really say much about Lyme testing, but the best way to determine if someone's been exposed to the pathogen is to feed a tick on them, because the tick will find the pathogen better than anything else. So most of the Lyme disease tests are serology tests, so test whether you have antibodies against the pathogen. If you feed a tick on a subject, that tick will pick up infection if it's present. So there were some scientists that were proposing to take clean ticks, feed them on people as a means of diagnosing whether those people had been infected. And as you might imagine, that went over not so well. So it never went too far. That's what happens when scientists are, yeah.
How do you think this graph applied to New York?
The New York ticks?
Okay,
New York ticks, is it? Sorry?
yeah, our tick patterns, they're gonna be the same like this.
It's the same. It's the same. I didn't show a map today, but there are two foci. There's the Northeast, including New York, Massachusetts, New England, and then the upper Midwest, Minnesota, Michigan, Wisconsin, and they both have the same pattern, these patterns. The Southeast, totally different story. The Southeast ticks, first of all, they see the reason for this cycle is they're actually a 2-year life cycle. So the ticks, these adult ticks will lay eggs that overwinter and then hatch out as larvae the next year. And then they come the next year as nymphal ticks. So it takes 2 years to complete the life cycle. In the Southeast, they have a 1-year life cycle.
What's causing the two rises here? The same tick or same population?
It is the same species. And we've speculated that maybe they were separate populations. Maybe there were 2 different cohorts, kind of like a cicada thing where they were off sync, but genetically they're identical. The thinking is that these are the one the ticks feed. These are ticks that didn't feed in the prior year. So it's just a kind of a carryover effect. We do know that these ticks are much less robust. So when we go out and catch them and put them in the containers and keep in the laboratories, these are hard to keep alive. These ones are pretty sturdy and robust. So it's almost like, yes, that they're just less, they're on their last legs, basically. Ticks, incidentally, they only move about a yard or maybe 2 yards in their entire life. They have very low metabolism. They take a single blood meal. They digest that blood meal to basically to mold to the next stage. So these ticks that have fed back here, by this point the next year, they're living off that single blood meal and their energy sources are very, very low. Yep.
How long could a tick go without the blood meal before we just die? Or are there ticks? Or do they also?
So this species can go months without a blood meal. So, basically there are ticks that start here and there are ticks that they're ending up in this part of the stage. Other species can go literally decades without feeding, not decades, a decade without feeding.
Okay.
These ticks aren't quite as hardy in that they don't estivate. So they don't basically go into a torpid state. Dog ticks do. So dog ticks will diapause. They can go into the leaf litter. They can shut down. They can conserve energy. These ticks don't have the ability to diapause. And so they have to sort of eat within a couple of months or they'll die. Anything else? Okay. I just got a few more slides here. Let me just show you a little bit more of the results here. So we can see things like where the ticks are biting people. Same point you made before about sampling. This is just where people are reporting ticks. So it may be that lots of people are getting bit on their butt and they don't find them, so we don't see them. But if you take this at face value, it seems that a large percentage of the tick come from the upper legs, the back of the head, the hairline, for those of you that have hair. Another interesting thing that's come out of these data, so this is something we published a couple of years ago. At that point, it was 51,000 ticks. It's now closer to 90,000 that we looked at. We looked at the distance because when people send us a tick, we say, tell us where you got bit by the tick and then tell us where you live. Because what we wanted to be able to do is communicate to the public about where the risk was occurring. Are you getting it when you're gardening? Or are you getting it when you go on your hike in the mountains where you drove a couple hours? So this is the distance between those 2 points of contact, where they live and where they get bitten by the tick. And you see that most of them, they're getting them in the same zip code. So they're either getting them in their backyard or they're getting them in the park that's around the corner. Although there are some people that are getting them from greater distances. And the reason that was of interest to us is, and we published this in Emerging Infectious Diseases, we found that lots of ticks that were getting reported around different parts of the country. In fact, most of the ticks that were getting reported in California were actually acquired while people are traveling in the Northeast or Upper Midwest. So most of the Lyme disease that people are getting is probably not endemic. It has travel histories.
Chart with a big arrow. That's the minute.
Yeah, that's them carrying the tick back to the place.
Welcome to New York.
Exactly, yeah. Our exports.
So can the Northeast ear tick survive in California? Was it brought out there?
So again, those are ticks, most ticks that feed on people don't survive, period. They might take a blood meal and fall off in your house, and they're probably not going to live in your house. So those ticks are probably doomed anyway.
So that even though they pitched a ride in California, they're not going to
Establish there.
establish.
So the way a tick would establish is it rides on a host that it feeds on and then falls off in the woods where it can get into the leaf litter, protect itself, and for the months that it takes the molt to the next stage. That doesn't happen in your couch or on the carpet or whatever in your house. They just die. I promised I would say a word about red meat allergy. So this is the oddest one of the diseases associated with ticks. There's a sugar that's in, I'm hesitant to say exactly where it is because I think there's still some debates about this, but it's been argued that there's a mimic of a sugar that's in red meat that's found inside the tick, whether that's derived from the tick itself or a pathogen that's inside the tick or a symbiont that's inside the tick is not clear. But this sugar induces allergy to red meat. So you get bit by a tick, you get exposed to the alpha-gal sugar, alpha-galactosidase, and you then develop an allergy to red meat. So you get bit by a tick, you eat a cheeseburger, you have an allergic reaction. And so hundreds of people now are suffering from this. And it was figured out to be a tick-borne etiology based on the fact that it occurred wherever it seemed like there were Lone Star ticks. And so again, it's not 100% clear that only Lone Star ticks can do this, but the predominant association seems to be between Lone Star ticks. So one of the things we hope to do in the future, so this is our distribution of human biting amblyoma ticks, Lone Star ticks from the country, is we'd like to be able to do some follow-ups with these individuals and say, okay, have you developed these? We can do some serologies, we can ask some clinical questions about whether they're experiencing difficulties with eating red meat, and try to drill down and figure out actually what the specific tick mechanisms are. Right now, it's not understood, the biology that is sort of the wild, wild west. In the interest of time, I'll just skip over that one. Yeah, so I guess I would argue that there are individual benefits. So we have a couple of anecdotal stories where pathogens were tularemia was detected in a tick in a child that the docs couldn't get the diagnosis. They saw the tick report and they're like, oh, well, we better treat for that, even though they're not detected in the serology. We actually have some published case reports for some of the rare pathogens like Borrelia and Miyamotoi. Again, the docs missed it. Parents had the tick tested, took the tick report. It was one of the case reports that was published was the mother had the tick tested. The father was a pediatrician, and his colleague was the attending pediatrician of this child that was developing pneumonia. The mother had the tick tested and insisted that they have the child sent to Yale University to another of our colleagues that eventually found that pathogen. But for the longest time, they looked at the tick report, presence of this pathogen of the tick, this kid with the illness that manifested all the symptoms of the disease, and wouldn't buy that it was actually, or wouldn't allow that might have been the case. But eventually they did, and the kid got better. Again, I thought there might be more, was thinking there might be a lot of public general audience here. So I was just going to point out that you can always go to our webpage. So if I said something that you don't remember or you had other questions, we have FAQs on our webpage. We have those shared statistics on our webpage freely open. And we also run a chat line. So we have people that are stand ready to ask questions about ticks and tick-borne diseases. The way this works is if you access it during the day, which basically means 9 a.m. to 9 p.m. on weekdays, you'll get a real-time answer. If someone's not manning the lines or there's not someone at the line at the time, it'll go to email and you'll get a response when they come back on duty, but generally within a couple of hours. And I'd also plug our colleagues at University of Rhode Island. They also have a great website if you're looking for more information about ticks. He sort of drills down a lot more than our page does about the different pet treatments and acaricides, and he's done a lot of the statistics or a lot of the studies about the essential oils and that kind of thing. So I encourage people to go to, it's called tickencounter.org. I guess this is the URL, but if you go to tickencounter.org, it just forwards to the Tick and Counter webpage. That's just my summary slide, and you can be tick safe. I think that's my last one. It is. Thank you very much for your time. Yes.
You said they're found predominantly along the edge, wood and field or something like that. 50% of the time they're there, or, because yesterday I was in the middle of the woods thinking, I'm walking through a whole mess of them, but the real problem is at the edge of the woods.
Yeah, because remember what they're doing is they're looking for something to walk by, and the edge is where they're going to most likely, and particularly white-tailed deer. So I would say 90% of them are on the edge.
So there is a vaccination for Lyme that dogs can take, essentially it's administered to dogs. Do you imagine this is going to be in the study? Do you imagine there's going to be a vaccination?
So there's actually a couple of vaccines for dogs. The one that's primarily used now is a recombinant OspA vaccine, which is the same OspA vaccine that was licensed for human use by two companies, and it was trialed and it had fairly good results, but they took it off the market back in 2000 because people weren't buying it enough, is what they claimed. Now there's, again, in the sort of folklore, there's stories that lots of people were getting sick from the vaccine and that's why they took it off, but there's really no evidence that that's the case. They argue that it just people weren't asking to get vaccinated with it. There are other measures going on now to try to develop new vaccines that target different things other than OspA, and we might see them on the market within the next couple of years. We're not involved in any of that work at present. One of the caveats I always have about the vaccines is it only protects against that one pathogen. Something we've explored, and actually lots of other groups are doing this too, is trying to look at whether you can develop vaccines against tick bites, not against the pathogens, but against the tick bites. So this has been done in cattle ticks. You can vaccinate cattle against tick bites by using a hidden antigen that's in the midgut of the tick, and then they develop antibodies that they imbibe with the blood meal, and it interrupts the feeding process. I'm trying to explore whether we can do that either as a wildlife vaccine that could be, broadcast as an oral vaccine to white-footed mice, or perhaps as a human or companion animal vaccine.
So kind of pre-infection kind of blocker.
Yeah, it would block all those pathogens.
Cows, maybe 7 or 8 years, that might be problematic.
Yeah.
So does the Borrelia burgdorferi cause any diseases to the white-footed mice?
No. It infects them, but they're asymptomatic. So it's probably evolution. They probably have endured this for quite some time and but it still affects it.
Okay, because cats would be similar. My wife and I have a vet clinic. We don't get cats with Lyme disease. We can get a lot of dogs.
So cats, I don't know that this has actually been demonstrated. I have a colleague that insists that cats are a risk factor for Lyme disease because cats are not very good tick feeders. They don't, black-legged ticks don't like to feed on cats. They get on cats, but they don't feed to reclusion, which means they get on the cat, they taste the cat, they don't like the taste of the cat, they fall off on the couch, and then they get on you, is what my colleague argues. So because of that fact that they don't probably feed as long, cats don't get infected with Borrelia at the same rate that dogs do, or even horses.
Okay, ticks are not on grass, it's apply only to the deer, right?
Ticks, sorry,
You said ticks cannot be found on grasses.
on
Grass.
generally not. That's all, I remember, I'll give you a story, like one time I told students, I was like, don't, we're out flagging ticks, the way you catch ticks, is you take a 1 by 1 square sheet of fabric and rub it on vegetation, the ticks grab on and grab them off. So I said, don't go to the grasses. Sure enough, kid went to the grasses and they got a bunch of ticks. So they're not, it's not that they're never there, it's just that they're predominantly in the edge.
But what you said is in reference to the deer, not other.
No, Lone Star, dog ticks and Lone Star ticks are in the grasses for sure. And that's partially because they can diapause. So the reason the black-legged ticks don't like the grass is that they can't maintain their body temperature and moisture as well.
One quick.
Yeah.
By grass, you mean mold.
I mean mold lawn.
Yeah, because, tall grass.
No.
Because they love it.
Do they prefer shade or sun? Or they don't care?
Ticks aren't very good at maintaining their water levels. They dry out. So in the sun, they'll dry out. So they like shady areas, which again corresponds to the edge, and leaf litter, where they can go in and they can sort of find micro habitats where it's moisture.
There is a West Coast species, a tick that is carrying Lyme as well.
Yeah, but yeah, it's called the Western black-legged tick.
And are they, would you say they're from the east and the west? Are we getting approached upon somewhere in the middle? Are they going to meet that reward of the world?
So here's that tick, and in our experience, they have very, very low infection rate. It's actually a different species of tick that has most of the Lyme disease there, but these ticks the reason is that most of these are feeding on fence lizards, and there's nothing in the middle part of the country. So the Rocky Mountains are basically a dead zone for these ticks.
And you imagine the state of that?
Yeah.
Do people ever send you soft ticks?
Very, very seldom. So soft ticks are, as the name implies, they're very soft integument, and they're usually nest parasites. So they feed on the nests of bats or in the Pacific Northwest, some rodent species. We've seen a couple in the past 3 years, but nothing before that. The Borrelia miyamotoi, that sort of rare pathogen I mentioned, that's a soft tick like Borrelia. It's usually not found in hard ticks, but it seems to be transmitted by hard ticks. So again, sort of a peculiar one. The difference being that Borrelia burgdorferi is not generally a bloodborne pathogen. It has a transient bloodborne stage, and then it makes its way into connective tissue. Borrelia miyamotoi is a true bloodborne pathogen like Borrelia hermsii or other soft tick Borrelias.
Huh.
I was curious about the mites because that you were seeing in the mussels. I bet they have a spirochete associated. They're big spirochetes.
There are some anecdotal reports of mites associated with diseases in freshwater mussels. But we really don't know that much about them, and the species have been poorly described. But basically with the freshwater mussels, we have this reoccurring problem where there are these spontaneous die-offs among one species or multiple species in the river, and people don't know if it's a chemical contaminant in the water or if it's a disease. And it usually appears to be specific to the mussels and doesn't seem to affect any of the other kind of aquatic wildlife.
Interesting. Well, thanks again for the invite. It's been a real joy to come up here and meet you all. Be happy to take more questions.